peace, shalom, hatap. Welcome back for chapter 6 of Balls of Fire, Spring Training 6, the cold reality of elite deceit. Don't forget to screenshot. It is imperative if you don't own the book and you want to read along. Please don't forget to like, comment, and subscribe. Your feedback is greatly appreciated. According to the texts of ancient Egypt, early China, and the 17th century American Navajo Indians, human potential depends on two ways of discarding energy: photosynthesis on Earth and chemiluminescence, the chemical reaction that dominates the universe. Whereas the chemical reaction of photosynthesis produces both light and heat, such as a burning candle, chemiluminescence produces light without heat or cold light. With their knowledge of science surfacing as mythology or storytelling, these early cultures explain how a death transition sets the stage for a cold light chemiluminescent transformation for human DNA through the operating ancient glycolysis pathway in our cells. This enzyme pathway has metabolic energy pathways for several sugars, such as glucose, maltose, and lactose, galactose, as well as lamporin receptor site for phage lambda. Consider that our DNA is composed of 4 main nucleobases enlisted as guanine, cytosine, adenine, and thymine. Chemiluminescence, the low temperature emission of light produced by chemical or electrical action, is characteristic of guanine, which can give rise to luminescence. The average G+C content of a 100 kb fragment in the human genome ranges between 35 to 60%, while phage lambda has a high G+C content of 49.858%. So emergence as a cold light chemiluminescent state of being may be possible through HGT and viral replication due to guanine content. If our DNA escapes degradation at death by microbes, water, and oxygen, scientists consider it somewhat immortal. Egyptologists today analyze mummified DNA that has escaped degradation. According to biologist Lynn Margulis, at death, we return to the bacterial world where freelancing DNA fragments hover between life and non-life, constituting a powerful toolbox for evolution. Microbes are our ancestors or planetary elders. More accurately, in Antonin Artaud's radio play entitled "To Have Done with the Judgment of God," he wrote, "Laugh if you like, what we call microbes is God." And side notes, photosynthesis is the reaction where a living organism captures the sun's energy, converting the light into water and carbon dioxide, then into carbohydrates, producing the byproduct of oxygen. Bioluminescence is a type of chemiluminescence where light and carbon dioxide are released by breaking apart organic materials using oxygen. And chemiluminescence is the emission of light by an atom or molecule that is in an excited state, whereas photosynthesis produces both light and heat. Chemiluminescence produces light without heat, cold starlight. The mesocosm. In between the two extremes of the microcosm, particles, and the macrocosm, humans, is the unexplored experimental gap of mesoscopic states, viruses and bacteria, essentially. As explained mathematically by Jasper Van Wezel, it seems reasonable that an energy or mass scale exists in the mesocosm between the classical macrocosm of large objects and the quantum microcosm of microscopic objects. In the mesocosm, dynamics occur at human time scale. In this then mesoscopic spectrum, state becomes equal in energy as the system grows to infinity. When this happens, it is possible to reduce to the system's quantum ground state, lowest energy state, while violating the system's governing physical law related to time symmetry. In 2010, for example, a violation of T, or time symmetry, means that time reverses due to quantum effects. In a magnetic field, time reversal symmetry can be broken by halting a system and allowing it to go backward on its previous trajectory. Ancient Egyptian texts describe the sun god coming to a halt at Osiris's mound and reversing time. Mesoscopic physics, a sub-discipline of condensed matter physics, deals with objects the size of a group of atoms, a molecule or material of micrometer size. Viruses and bacteria are the systems normally studied, and these objects are subject to the rule of quantum mechanics, not the classical physics that governs our physical world. Actually, at this mesoscopic scale of molecules, DNA transcription and translation is occurring, and DNA fragments survive due to self-assembly with other molecules or gene transfer via bacteriophages, such as the very abundant phage lambda. In this quantum domain, self-organizing principles are at work, such as crystallization, as Egyptian, Chinese, and Navajo eschatologies support, relative to a complex process involving protein synthesis and non-native interactions resulting in degradation. The marriage of elite deceit and altruism. Regarding the survival of our species, human history can be explained as a non-conscious physics game of political deceit about knowledge determining whose DNA will survive or transform at the transition of death. As you shall see in the pre-game of the game of centuries, ideologies, religion, physiology, capitalism, and so on promote an underlying biological altruism, which is self-sacrifice, that undermines the individual's power through normalization processes. So historically, chemical knowledge of human transformation to a crystal or morning star was mystified, satanized, literalized, criminalized, and controlled by mechanisms of power. However, many historical individuals understood human potential for a cold-like crystallized transformation. For instance, American philosopher and mathematician Charles Sanders Peirce envisioned the world becoming absolutely perfect, rational, and symmetrical system in which mind is at last crystallized. Peirce said that man should become welded into the universal continuum to prepare himself for a transmutation into a new form of life, what suggests an emergence to a higher order crystal state of being. According to one study, the 98% microbial DNA in our cells possess the potential for some as yet unspecified goal. After all, the history of life and non-life exists within our cell. The science explains how our journey began with nature's cosmic logic of star birth, chemiluminescence, followed by a carbon-based life on Earth, which is photosynthesis. Bioluminescence is the chemical reverse of photosynthesis. The ISIS thesis proposes that a creation process involving protein folding continues to cycle via the chemical reaction of bioluminescence or chemiluminescence because our large microbial DNA toolbox has the potential for an emergent creation rather than DNA degradation and human death. Degradation is when the steady state level of a protein is reduced. Proteins in cells are subject to stress due to glucose starvation, heat shock, and mutation. Oxidative stress damages many proteins, making them genetically unstable. To use these proteins are targeted for destruction. Degradation or proteolysis is the mechanism that salvages unusable amino acids from non-essential proteins. However, the mechanism of horizontal gene transfer allows a DNA fragment hovering between life and non-life to enter a cell for DNA transcription and translation. So it is not unstable, not stressed by glucose starvation, and not degraded by oxygen, water, or microbes. So, in the mesocosm, it may be possible that a bacteriophage, ferry boat, or carrier molecule might transfer human DNA to avoid degradation at death transition. One must know where to find a viral ferry boat, and this is what ancient Egypt, early China, and the Navajo are explaining in their eschatological literature, how to find a viral ride to avoid degradation. It is like trying to find a taxi home on a busy night after a ball game. If you don't know exactly where to find a taxi, you are out of luck. Also consider that there is room in the taxi for only one DNA fragment to be horizontally gene transferred from transformation due to vector requirements on the lambda genome. Add to this crystals and DNA have atomic spatial limitations, and you are beginning to understand why the pharaonic elite kept chemical information away from the plebeians. Put simply, there are not enough DNA seats in the viral DNA taxi or the resulting crystal. So this results in non-native interactions and the degradation of DNA. Now, in the case of lambda, C1, and crow proteins, their adjacent genes on the genome allow their folding funnel landscapes to interconnect in their binding battle for genetic seats, or gene seats. With extreme simplification, when two stable states exist, the underlying energy landscape rotates similar to two vortices in opposite directions, or a vortex/anti-vortex pair. In this case, dissipation is essential, and without it, the system cannot change its energy. As you shall see, dissipation or degradation is essential in the lytic lifestyle of phase lambda, where two competitive protein exhibit two stable states that function similar to a rotating black hole with two horizons or a vortex/anti-vortex pair.